Hello, and welcome to this Edison webinar, the CNS Renaissance Biotech Companies at the Forefront. My name is Aaron Atkar. I'm a healthcare analyst here at Edison. As I'm sure you're all aware, while a significant portion of our focus is on the bottom-up work that we do on individual stocks, we also consider the broad themes related to the companies we work with. And in September, we published a thematic piece on the CNS Renaissance, which, if you haven't already, I'd encourage you to go and read it. In the meantime, though, what's exciting about today is that we've got 4 innovative companies that are developing next-generation CNS therapies. So the format of the event will be company showcases, starting with a focus on neurodegenerative conditions. We have Herantis Pharma and Alzinova. We will then move on to those with more of a focus on neuropsychiatric conditions, where we have Compass Pathways and Newron Pharmaceuticals. Each company will give a 10-minute presentation, and then we'll then follow that with 5 minutes of Q&A. We've had some questions come in via email already, which is great, but we'd also like to encourage our audience to send in questions throughout the talks via the chat and Q&A function at the bottom of the screens. That's enough from me, though. I'll now introduce our first speaker. So from Herantis Pharma, we have CEO Antti Valanta, who has a vast experience in financing, partnering, research, as well as the development and commercialization, manufacturing of biological drugs. His showcase today will focus on Herantis Pharma's recently announced positive trial results in Parkinson's disease. So Antti, over to you.
Thank you, Aaron. Welcome also on my behalf. So Herantis Pharma is a Finnish clinical stage biotech company, and we have ambition to stop the progression of Parkinson's disease. So next slide, please. So the forward-looking statements there, and then a summary of Herantis Pharma. So our lead asset, as we call it, is HER096, which is a small peptide, brain-penetrant peptide that activity is derived from a neurotrophic factor called CDNF, and this makes Herantis quite unique, as we already have evidence on its clinical characteristics and biological activity in Parkinson's patients. And based on that study, we then developed HER96 as a brain-penetrant peptide having the same mechanism of action as the neurotrophic factor CDNF. And we just completed a phase 1b clinical trial with subcutaneous HER096, and that was the study with first time studying Parkinson's patients in clinical studies. And we were very happy about the results of that study. And of course, now we are setting up the phase 2 efficacy trial. Next slide, please. So HER096 mimics the CDNF neurotrophic factor neuroprotective activity, and we address a pathway called unfolded protein response pathway, which resides in the ER of the neurons, and that pathway and the role of that is to address cell stress by reducing misfolded and unfolded proteins in the ER. And normally it's a homeostatic process in the cells. However, in pathological conditions like Parkinson's disease, the ER stress is constantly activated, and that drives neurodegeneration and eventually also cell death. The natural role of CDNF is to control this ER stress or unfolded protein response pathway by bringing back the normal homeostatic process. And what we did was resolve the structure of CDNF and its binding partner GRP78. GRP78 is the compound that, or the ligand that actually controls the unfolded protein response pathway. And we identified the binding interface of CDNF. On the right-hand side figure, you can see that pink residue representing that binding interface. And we developed HER96 based on that binding interface. So HER96 is a mimic of the binding interface. and it has nice characteristics as it's very efficiently penetrates also the brain. Next slide, please. And this slide now summarizes what I already mentioned about the HER96 coming or derived from the CDNF protein, and we have shown in preclinical settings that yes, we can capture the full activity, the neuroprotective activity of the CDNF protein. So basically, we can reduce cell stress. We can also address protein misfolding and reduce neuroinflammation. So basically, we can protect the dopamine neurons from further degeneration, and we can support their functional restoration. Based on preclinical settings, we also know that we have fairly efficient and quick symptomatic improvement because we are able to induce the dopamine production by the affected neurons. But of course, we then want to achieve a long-term effect with disease modification, so slow down or even stop the process of the neuron degeneration. And what we currently believe is that subcutaneous administration twice a week is the right administration regime. Next slide, please. So how we describe the challenge of the degenerating dopaminergic neurons. We describe that as a vicious cycle composed of the accumulation of toxic alpha-synuclein aggregates, the ER stress, and neuroinflammation. And in this vicious cycle, all of these three components strengthen the effect of the others. And of course, this results in decreased dopamine secretion, so degeneration of the neuron and eventually the death of the neuron. Next slide. However, HER96 can rescue the neuron function, so we target the unfolded protein response pathway simultaneously in the neurons, but also in the glial cells. So we can break down this vicious cycle by actually having effect on all of these key characteristics, so on protein aggregation, on unfolded protein response pathway, so ER stress in neurons, and also in the glial cells, which results in increased dopamine secretion and of course, and then thereby normalized motor function. Next slide, and the following one. Yes, thank you. So we do have strong evidence, of course, about HER96. First of all, with CDNF, the neurotrophic factor, we ran a comprehensive preclinical program and also a phase 1 clinical trial in fairly advanced Parkinson's patients, where we actually saw in several patients both clinical responses and biological responses within the 12-month study, where CDNF protein was administered intracranially in Parkinson's patients. Next slide. With HER96, we have, of course, the improvement that we can use subcutaneous administration delivering the same activity. And we have very robustly shown that, yes, we captured the full activity of the neurotrophic factor with subcutaneous HER96. Next slide. And in this slide, we can clearly demonstrate the previous one. Okay, we can't see a video here that actually shows the neuroprotection. So basically, we have been able to improve the motor functions of aged mice, which should have been shown in the video. And of course, in clinical settings in phase 1b, we have shown efficient blood-brain barrier penetration, very good safety profile. We have selected those for phase 2 clinical trials. We are still waiting for biomarker data from phase 1b from patients. We expect to report the data by end of the year. And there our ambition is to show the effect on the target biology in the CNS. So next slide. Our phase 1 program composed of 2 studies. First of all, we ran a single ascending dose study in healthy volunteers, where 60 healthy volunteers were injected with HER96. And in that study, we also had elderly individuals from which we were able to take CSF samples, where we demonstrated a robust blood-brain barrier penetration. And now in the recent phase 1b study, we completed pharmacokinetic studies in terms of BBB penetration and dosing interval. And based on that, we were able to select the preferred dose for phase 2. That would be 300 milligram dose twice a week. And then we had 24 Parkinson's patients, early to mid-stage patients at 2 different dose levels who received then HER96 doses twice a week for 4 weeks. And as a conclusion, we had very nice safety profile, no systemic adverse events, some adverse events related to actual subcutaneous injection as expected. Next slide. And as already mentioned, our objective and ambition is to demonstrate the biological response to HER96 dosing in the biomarker sample analysis that will report by the end of the year. Next slide. And the next, yes. So as related to company financing or funding, very delighted that the Michael J. Fox Foundation and Parkinson's UK actually fully supported the phase 1b clinical trial, and also some other biomarker activities with almost 4 million research financing. In addition to that, we do have an investment commitment from EIC fund, so they can invest equity into the company up to 15 million, and from that reserve we have utilized so far 3.2 million. So basically, in case the company raises additional financing, they are able to chip in 1/3 of any subsequent financing round up to the remaining 11.8 millions. Next slide, please. And of course, our ambition currently is to set up a phase 2 efficacy clinical trial in early to mid-stage Parkinson's patients. We expect to start this trial by or during the second half of next year. And in addition, we are looking at pharma partnering, and of course we have discussed with pharma over the years, and it seems like that the latest results with HER96 are really nicely resonating within the pharma industry. So that's of course a great to see as a CEO of the company. Next slide, please. So this basically concludes my presentation. So just to in a way emphasize that we believe that HER96 is a potentially game-changing therapy that could become the first disease-modifying treatment for Parkinson's disease. All right, thank you.
Fantastic, Antti. Thank you very much for the presentation. Sorry we couldn't get that video to work, but I would encourage everyone to go and have a look at it. It's a really nice visualization of the effect of HER96. Just as a reminder to our audience, we've got the Q&A function in the bottom, so please do feel free to send in your questions throughout the talk. Antti, I wonder if I could ask you one to get going. It's been great to see this novel mechanism of action for HER96 and see its application in Parkinson's disease. I think you sort of touched upon its potential to expand to other disease areas. I wonder if you could elaborate a little bit on that. Are there any in particular, CNS or otherwise, that jump out?
Yes, absolutely. So we are addressing a fundamental pathway that is driving neurodegeneration. And of course, that pathway is not specific for Parkinson's disease. And actually, with the neurotrophic factor, there is scientific literature about effects on in Alzheimer's and ALS, Huntington's, and actually also in stroke models as well. However, not only neurodegeneration, but it's that pathway, unfolded protein response pathway, is really relevant also overall related to aging. So we believe that there are many opportunities also outside of CNS. And of course, the company's ambition going forward is to start really addressing these other indications. Of course, for that, we would really much like to partner with pharma, the Parkinson's program, so that we would have the resources to really dig into other indications.
Excellent, thank you. We saw in the presentation then that there's currently no disease-modifying therapies available for Parkinson's, with few in development. Could you elaborate perhaps on who else is working in the space? And are there any other sort of companies that investors should be aware of that are taking a similar approach to treating the underlying disease rather than symptoms?
Yeah, of course. I think the most advanced programs are related to removal of alpha-synuclein aggregates by immune therapies, and there maybe most advanced one is Roche, who is forwarding their program into phase 3 next year based on the phase 2 results that they have published during the past a year or so. And then there are also other companies. Many of phase 2 companies are actually addressing a subset of Parkinson's disease. So based on, for example, genetic modification, good to emphasize that in Parkinson's disease, there are quite limited amount of genetically defined diseases, maybe between 5 to 10%, and we as Herantis, we believe that our HER96 can really address very broadly, like sporadic Parkinson's patients. So we have no reason to believe that we should somehow limit eventually the patient population.
Excellent, thank you. Maybe just one more question for you, Antti. Seen that the full biomarker data sets from the phase 1b trials available hopefully before the end of the year. So for those without a scientific background, perhaps, could you explain what you're hoping to see in that data that would give you confidence that the drug is working at a biological level, and then maybe sort of what you'd consider to constitute a positive result that would de-risk investment ahead of phase 2?
Yes, so phase 1b clinical trial, there we injected HER96 in Parkinson's patients for 1 month, and 1 month is really a short time period to demonstrate disease modification in Parkinson's disease, as Parkinson's disease is really a slowly progressing disease. So what we want to show in biomarker data is that yes, we have been able to affect the target pathology, so the unfolded protein response pathway, and that would be in a way the triggering a series of events that we believe that will eventually turn out to be or result in improvement of the motor symptoms of the patient. So in a way, starting the cascade that eventually results in efficacious treatment. So we believe that will be a really important milestone for the program. And as you mentioned, it will really much de-risk the phase 2 efficacy trial.
Fantastic. Antti, thank you very much for your time today and for the talk. That was really interesting. We're going to move on now to our second presenter. We've got Alzinova, and we have CEO Tord Labuda, who joined Alzinova in 2024, bringing over 15 years of experience and leadership roles within the pharma industry. So Tord's showcase today will give a brief introduction to Alzinova and the market it operates in, highlighting how it aims to improve quality of life for Alzheimer's patients. So over to you, Tord. Thank you.
Thanks a lot, Aaron. Great to be here, and looking forward to talk a little bit about the company. So our mission here is to make it possible for Alzheimer's patients to live an independent and active life. We can go to the next slide. And the next one. So Today, we have about 55 million people living globally with Alzheimer's disease, and we are expecting these numbers to double over the next 25 years to more than 100 million people by 2050. Next slide. This is, of course, devastating, but it also brings a great market opportunity. We have a CAGR right now with 8 to 12% growing, and the potential of annual sales in excess of 12 billion for any true disease-modifying treatment. And we can move on to the next. So before talking about our compounds and our pipeline, just a little bit about Alzinova. So we're a Swedish-based biopharma company. We're based in Gothenburg and was founded in 2011 as a spin-out from the University of Gothenburg. We believe we have a very innovative science and an IP technology that makes it possible for us to make or create disease-modifying treatments for Alzheimer's disease. I'm going to talk about two things today, primarily about our therapeutic vaccine candidate, ALZ-101, that is phase 2 ready. We have recent phase 1b data that I will talk a little bit about. We also have an antibody, ALZ-201, towards the same target that is in the preclinical state. We believe that we will be able to bring this into the clinic over the next year. We're listed in Sweden on Nasdaq First North, and we have about 5,500 shareholders. Next slide, please. Next, please. Yeah, this one. So with the treatment, we have basically two new disease-modifying treatments on the market today, Leqembi and Kisunla. They are both IV infusions. So this means that the patients need to come into the hospital and being treated up to 26 times per year in a clinic or at the hospital, which of course is very cumbersome for the patients, for their accompanying family members that need to bring them. And then of course it comes with a tremendous cost to society. And we are looking at about $15,000 per patient and year. And this is excluding the cost of the actual drug. With our treatment, we are looking at approximately 3 injections, simple intramuscular injections that can be done at 10 minutes at any clinic or hospital, even in primary care settings or at home. The cost for society here will be a fraction of that, estimated to about 2%, meaning that we can actually treat 50 patients for the cost of one. I should also, of course, mention that the current treatments, such as Leqembi, are going into subcutaneous versions now. But that comes after a long period of 18 months on IV infusions. And on top of that, the actual subcutaneous treatments are to be taken weekly. So it's still very cumbersome and costly treatment options. Yeah, we can move on. So our business model is based on, of course, develop these treatments through clinical trials, most closely in the time now going through the phase 2 program here. We of course want to document efficacy, safety, and tolerability in these patients, and then our aim is to partner with big pharma. I should mention we are in discussions with a number of companies right now, and have to talk to almost everybody that are involved in CNS of the big pharma companies. We believe that we can have a license deal here with incomes through upfront milestones and royalties. So next slide, please. So what is it that we actually do, and what does our candidate look like? Next slide. So I'm sure many of you are familiar with the A-beta pathway or amyloid beta pathway, which is based on amyloid precursor proteins that can aggregate in different ways and ultimately form these plaques that we have known exist in the brain for more than 100 years now. These plaques are insoluble structures that occupy a big space in the brain, actually. And we do have 2 treatments now on the market, as I mentioned, Leqembi from BioArctic Eisai Biogen, that are focusing on removing the plaques, but also claim to bind other forms of this amyloid beta precursor proteins. We also have Kisunla from Lilly that primarily are removing the plaques. However, what we see from the studies is that despite the fact that these treatments removes the plaques, this doesn't translate very well into clinical efficacy when you look at the cognitive behavior of the patients. At the same time, we have known now for a long time, 10 to 15 years, that there's something else that is much more toxic to the neurons in the brain. And this is A-beta-42 oligomers, or a specific type of A-beta-42 oligomers. These are soluble, very unstable structures in the brain that are extremely toxic. We have shown now in many preclinical models in vitro that by removing these neurons, these toxic oligomers from extract from Alzheimer's patients, the extracts becomes much less toxic to the neurons. So we can move on to the next slide. So what we have done with these molecules is that we have stabilized it by inducing a disulfide binding in the monomers, and then stabilize it in a way where we can actually use it for drug discovery. We are inducing this together with an adjuvant, aluminum hydroxide, of course, and this is a simple intramuscular injection that I mentioned. It will make the body then, of course, produce antibodies towards these toxic oligomers. It has been designed so we can have a long-lasting effect with very minimal booster dosing, about 3 times per year, looking at the phase 1b data we have now. Because of this, we believe we will have a very high patient compliance, and it also makes it possible for us to have a preventive treatment here, or a prophylactic treatment, if you will, which of course will be great once we can identify patients that later in life will develop Alzheimer's disease. And I think that the recent data we have from the biomarker studies now, which very well diagnose patients with Alzheimer's disease, will in a few years now also be possible to actually predict who's going to develop Alzheimer's disease. So with this, we will address a substantial global population here. Next, please. As a complement to the therapeutic vaccine, we also have ALZ-201, which is a monoclonal antibody also targeting these toxic oligomers. We believe that this could be a powerful combination strategy together with the vaccine candidate, but also a standalone treatment. We believe that it could be extremely valuable also for populations that have, for some reason or the other, have a difficulty to mount a sufficient immune response, such as immunodeficient patients or very elderly patients. This antibody, since we are so specific towards the oligomer, we are not targeting the plaques or any other forms of A-beta. Means that we will also be able to have this in much lower concentration, which also makes the blood-brain barrier not a huge hurdle for us. We will get sufficient antibody concentrations into the brain without using brain transporters and the likes. Okay, next slide, please. So this is the phase 1b trial that we have performed. We started out with 26 patients with early AD or mild cognitive impairment, 3 arms, placebo, 125 and 250 micrograms. The patients were injected 4 times at 0, 4, 8 and 16 weeks, and then we had a 48-week follow-up period. The patients that could and wanted to could move into part B, which was open label, and 4 additional injections over the same course of time. So in total, the patients that were on active dose through the whole study were in the study for about 2 years. Once we saw the treatment was safe, we also had an additional arm with 6 subjects that got a higher dose to check if we could have higher immunogenicity levels, basically. So the primary endpoints here was looking at safety and tolerability. As I mentioned, immunogenicity, and explorative endpoints, we had biomarkers both in CSF and blood, and we also looked at cognitive in many different scales. The main one was ADCOMS, which is the same scale that Eisai used for the Leqembi studies. So moving on to the next slide with the results. Unfortunately, I can't show you the graphs now. The data here will be presented in detail at CTAD, Clinical Trials in Alzheimer's Disease in San Diego next week. So we have 2 posters there that will be presented. But the overall data is that on the safety and tolerability side, we have shown that we have a very safe and tolerable drug here. On the immune side, we see a very robust, long-lasting and importantly, recurrent immune response indicated that we are able to have a memory B cell response form here, and we see this in more than 90% of the patients with expected IgG levels also crossing over the blood-brain barrier and being present in the CSF. For the explorative efficacy, looking at the cognitive scales, we really see that the patients that are on active treatment throughout the study don't progress in their disease, indicating that we are able to halt their disease progression. This is in turn supported by biomarkers, and primarily a biomarker called neurofilament light that has proven to be a very difficult biomarker to move with the other treatments that are on the market. They don't have an effect on this biomarker. NFL is known to be involved in many neurodegenerative diseases as a marker of neurodegeneration, including Parkinson's disease. And we can see that this biomarker moves in the right direction in our trial, in the same direction as the cognitive data that we see here. We also have an additional very interesting finding in our study, and that is that we can see when we look at healthy elderly individuals, we can see that these people have naturally occurring antibodies towards our toxic oligomers. When we then look in young people that are far from developing Alzheimer's disease, or they have very low levels of these antibodies. Similarly, when we look in the Alzheimer's patients before being treated, they also have very low levels of naturally occurring antibodies towards the oligomer. However, when we vaccinate them, they come up to about the same levels as the healthy elderly that in levels, which indicate that we are looking at Alzheimer's disease potentially being driven by an immunodeficiency. So this is something that we are looking into a lot more now. We also have some. We also looked at some adverse events of special interest. Of course, we have seen with the other treatments that they are inducing ARIA events or edema and microbleedings in the brain. In our hands, we don't see anything on microbleedings area H above background levels, and on the area E events we only had one event, and that came very late in the study, and in a patient that came from the placebo group, and we believe that it is not related to the drug. So that's the phase 1b data. Next slide, please. So what are we doing now is, of course, advancing this towards phase 2. We just have our IND cleared here a few months ago, and the fast track less than a month ago. We are progressing with the EMA submissions, study design and protocol are of course in place, and we also have selected the CRO that we will work with, Worldwide Clinical Trials. We are continuously developing our drug towards the study start, and at the same time we are further strengthening our IP portfolio. So the path here is to find a partner moving this forward, and this is discussions that have been ongoing for quite a while now, and we are advancing the talks with a number of parties here. Yes, next slide please. So this is a little bit about the timelines. We are initiating our phase 2 trial as soon as possible and pending financing, of course. Alzheimer's is an expensive field. The trial we are going to perform is about 240 patients, and we believe that we will be able to have a halfway interim readout after 12 months into the study, and then the final study report in 28-29. Thereafter, we will move on to phase 3. Next slide, please. So this is just a summary of what I've showed you already, and I think we go to the next slide and present the team real quick before I stop. So Anders Sandberg, as mentioned, co-founder of the company, is still with us as the Chief Scientific Officer. The rest of the team have everybody been involved in taking drugs to the market before, and we believe that we will certainly be able to do this with this fantastic opportunity we have at our hands here. So next slide. Thank you.
Excellent, Tord. Thank you very much for the presentation there. I wonder if I could ask you a quick question to kick things off here. Seeing that your work appears quite unique to me anyway, and that it's sort of immunotherapy, but for a sort of CNS indication. So given that immunotherapy is sort of usually associated with immuno-oncology, be good to hear your thoughts on why immunotherapy approaches are sort of a bit more generally underexplored in CNS conditions.
Yeah, I think that, first of all, I think it's a breakthrough really that we have the first disease-modifying treatments in Alzheimer's disease, and I guess it's an immunotherapy, yeah, both Kisunla and Leqembi, although it's passive immunotherapies, right? So I think that this is the next step, and I think the reason we have done it in oncology is that there is room for personalized treatment in a different way, right? And in that sense. That said, we don't really have moved down the vaccine route in cancer either yet. So that remains to come also for the future. But I think in this sense here, we're looking at, inducing antibodies or giving them passively. In our case, we believe that doing this by stimulating the immune system will be as effective as actually doing this by a passive immunization, because we are very specific. We don't need super high levels to come in bulk over the blood-brain barrier. I think that we will sufficiently over time have when you look at the area on the curve over time, I think we will have very good levels of antibodies in the CNS. So I think it will be a great way of treating Alzheimer's disease, actually.
Makes a lot of sense, thank you. We've had a question come in asking if there's potentially any path to an accelerated approval if you were to see strong phase 2 results.
Yeah, I think, the fact that we have the fast track approved, that is a huge thing for us. Not only does it confirm that what we believe, that our data is strong enough to move down this road, but it will of course also help us in our interactions with the FDA. And going forward through phase 2 and hopefully phase 3, we will have close interactions with them, and this will hopefully also include accelerated approval, expedited review of our data, and hopefully we can also use part of the phase 2 data we have as pivotal data going forward.
Thank you. With ALS 201 in preclinical development alongside the 101 lead candidates, could you explain a bit more about the sort of rationale behind developing both in tandem? Is this two separate shots at goal, or could they be used together perhaps? And maybe could you just remind us again on the key difference between those two candidates?
Yeah, right. So the vaccine is stimulating the immune system, of course, by itself. There is little evidence, of course, that this will be an effective therapy for a neurodegenerative disease so far, except for what we have generated. I would say that we know now that passive immunizations work, at least to some extent, right? So ALZ 201, you could say, is a safety backup for potential failure. But that said, we also see that it's common that very old people, say in their 90s or so, might have difficulties to develop an immune response towards something where you try to stimulate it with a vaccine, right? We see that for other vaccines. So having the 201 as an alternative and maybe booster their immune system might be a good thing, right? And then, of course, we have patients with different type of diseases, like HIV, for instance, where you definitely will have challenges in mounting an efficient immune response. Their passive immunizations will be the way to go, I think.
Excellent, Tord. Thank you very much. That wraps up the first part of the event, where we've sort of covered two exciting companies focused on neurodegenerative diseases. We're going to shift the focus now onto two innovative biotechs focused on addressing unmet needs in neuropsychiatric conditions. So the first of those two companies, we've got Compass Pathways, where we have Chief Patient Officer Steve Levine and Senior VP of Investor Relations Steve Scholz. Steve L. is a board-certified psychiatrist who has spent his career working across multiple facets of the healthcare system to improve people's lives through creating access to innovation. Steve S. has an extensive background in investor relations, corporate communications, and capital raising. Steve was actually previously based at Newron Pharmaceuticals, the company that developed the first FDA-approved cannabinoid medicine, Epidiolex, for epilepsy. So I'll hand now over to the two of you, Steve and Steve.
Good. Well, thank you, Aaron, and thank you, Edison, for having us at this conference. And before we begin, we will be making forward-looking statements, so we point you to the risk factors listed in our SEC filings. Compass is listed on the NASDAQ exchange under the ticker symbol CMPS. We only have one slide today, but two speakers, so a different balance here. For a more detailed investor presentation, please visit the investor section on our website. Compass Pathways is a mental health company. We're focused on bringing innovative options to patients that suffer from mental health conditions of significant unmet need. And our first product is Comp 360, which is a synthetic proprietary psilocybin treatment targeting treatment-resistant depression, or TRD, and PTSD. And we have breakthrough designation for TRD, which offers an accelerated approval pathway. We are currently in phase 3 for TRD with 2 trials. The primary endpoints are at 6 weeks, and the trials are blinded to 26 weeks. The first of which we call 005 compares a single administration of the target dose of 25 milligrams versus true placebo. The primary endpoint for this trial read out in June of this year and was highly statistically significant and well tolerated from a safety standpoint. The second trial we call 006 is 3 active arms, a subtherapeutic dose of 1 milligram, a mid-dose of 10 milligrams, and the target dose of 25 milligrams. And in this trial, we're giving a second administration within the 6-week primary endpoint period. The design of this 3-arm trial, the 006 trial, is the same protocol as a phase 2 study that read out several years ago, which was statistically significant at 6 weeks, highly statistically significant at the primary endpoint of 3 weeks. And the primary endpoint of 006 is due out alongside the 26-week 005 data later in Q1 next year. With these readouts in Q1, we're looking to see how a 2-dose administration compares to a single dose. And as we look at the 005 26-week data, which we'll see again at the same time, we're looking for trends that will inform commercial utilization. Mainly what the expected frequency of administration will likely be, and Dr. Levine can go into more detail on that as well. We also expect to announce a single late-stage PTSD trial soon, and we have high conviction in PTSD for Comp 360 due to an earlier open-label study of 22 participants that was very promising both from a safety standpoint and from a treatment effect. And we expect that trial to start early in the new year. We have had very positive and productive interactions with the FDA, and as we announced on our past quarterly call, we have accelerated our commercialization efforts and now expect to move our commercial launch forward by 9 to 12 months. So pretty significant move forward, and that should bring with it pretty dramatic value. With that, let me hand the call to Dr. Stephen Levine, and he can provide a bit more detail on the unmet need in TRD and PTSD, our clinical programs, and importantly, our commercialization initiatives. Steve?
Thank you, Steve. And by the way, you don't have to be named Steve to work at Compass Pathways, but it doesn't hurt. At Compass Pathways, we are committed to developing new options to transform the lives of those living with severe mental health conditions. We can become numb to the statistics, but the size of the unmet need is truly overwhelming. There are more than 3 million US patients suffering from treatment-resistant depression, which is a major depressive disorder for which someone has been failed by at least 2 treatments within the current episode. And this represents a tremendous burden on patients, their caregivers, and the healthcare system. In treatment-resistant depression, there are currently only 2 approved pharmacologic options. Really, only one of them is actively prescribed with any volume, and that is Spravato, which was approved in 2019. In 2024, Spravato exceeded $800 million in sales worldwide. They're on track to exceed $1 billion this year, but it's important to point out that that represents in 2024 only about 60,000 patients out of, as I mentioned before, the more than 3 million living with treatment-resistant depression. As a psychiatrist who has treated thousands of patients with TRD, it is dismaying to realize that the reality is that most patients living with TRD languish being prescribed treatments that have not demonstrated efficacy in this disease state. There are more than 50 approved treatments for MDD, and many of those drugs have been tested in a treatment-resistant population and have failed to show efficacy. So again, we're talking about a very large number of patients who have very few options today, and the options exist often result in tremendous burden on their patients and their caregivers in terms of lack of durability, the frequency of treatment, and what is required to actually access those treatments. As Steve mentioned, we also have a program in PTSD. PTSD is another area of tremendous unmet need, with approximately 13 million US adults each year suffering from this condition. There are also limited effective treatment options in PTSD. There are only 2 approved pharmacologic treatments. There hasn't been an approval in PTSD in decades. The existing approved options are old generic SSRIs that have moderate efficacy. And so this is a group of patients who really right now are underserved with existing treatments. Based on the data we've generated to date in COMP360, we are very encouraged that we have the potential to deliver a rapid, durable, and meaningful set of improvements for these patients. Steve covered some of the highlights of our development program to date already, but I'm going to dive into some more detail. We do have active development programs in both PTSD and TRD. Starting with TRD, the active trials are our 2 ongoing phase 3 studies that we have named COMP005 and COMP006. Starting with 005, this is a single administration prior to the primary endpoint at week 6, a monotherapy study comparing 25 milligrams to an inert placebo. As Steve said, we've already reported the top-line data from this study. This was in June of 2025, and we were able to demonstrate a highly statistically significant result with a clinically meaningful difference in the change from MADRS at baseline to the primary endpoint at week 6 following just a single administration, comparing that 25 milligram dose to the placebo. Not only was this highly statistically significant, it was clinically meaningful, and we believe highly commercially viable, particularly given the fact that we were able to demonstrate durable response at week 6 after just a single administration relative to any other treatment available today that is already highly differentiated. COMP006, as Steve already mentioned, we'll also look at our 25 milligram dose of COMP360, but versus 2 active doses, 10 and 1 milligram. And different than our phase 2b study, which I'll cover in a moment, this will look at 2 initial administrations of COMP360 at a fixed interval, 3 weeks apart, still with the primary endpoint at week 6. We will have part A, that the initial endpoint plus the key secondary endpoint for this study available alongside of part A from 005 and the 26-week data from 005 in late Q1. And we look forward to being able to compare 1 versus 2 initial administrations of 25 milligrams to see if that second administration elicits a deepening of response or more responders. Preliminary data suggests that we met. Along with that, we have already demonstrated robust data from our very large phase 2b study. The results of this were published in the New England Journal of Medicine in 2022. This was a study in 233 participants in 22 sites across 10 countries. We saw a highly statistically significant result at our primary endpoint, which was week 3, comparing our 25 milligram dose to a 1 milligram active comparator. We also included a 10 milligram intermediate dose in this study. This again was measuring the change from baseline to that primary endpoint with a single administration. And for those who did meet criteria, who were responders or remitters at that primary endpoint, the vast majority of them sustained that through week 12, the end of that study. Important also to note that the primary endpoint at week 6 in our first phase 3 study, COMP005, was very comparable to the same time point in the phase 2b. It was a very consistent result. Also important to mention that along with the report of the top-line efficacy data in the 005 study this past June, we also had a statement from our independent DSMB that was reporting on safety data they reviewed both from 005 and 006 up to that point in time, which surpassed even just the primary endpoint of 005 at that point. Many patients had progressed further in those studies. And the DSMB reported that there were no new safety signals and there was no clinically meaningful difference in suicidality across the arms of that study. So a very favorable safety statement from the DSMB. With the upcoming data readout in late Q1, we will also have additional safety data from Part A that will include full adverse events, but we'll also have a chance to see longer-term safety with the inclusion of Part B in that readout. Following the readout of the top line from 005, we had a very positive meeting with FDA, which included alignment on a rolling submission after we have additional data available in late Q1. This is to enable an accelerated NDA filing for COMP360 in TRD. We will look to accelerate as quickly as possible, again, recognizing the tremendous unmet need, while also staying committed to the same standards of efficacy that would be needed for the approval of any new treatment in major depressive disorder or treatment-resistant depression. Moving on to PTSD, we had a phase 2A study that we read out last year and has been peer-reviewed and published. This was an evaluation of our 25 milligram dose open label of COMP360 in a population with PTSD. This was primarily a safety and feasibility study, and this was a very well-tolerated treatment by these participants. And this was 22 patients open label. There was also an efficacy endpoint that generated a very exciting signal. So we are very motivated, very excited to progress the exploration of COMP360 and PTSD. And as Steve mentioned, we are looking to kick that study off in early 2026. So with that, I will hand back over to Steve Schultz to talk about cash and other preparation.
Thank you, Steve. So in conclusion, we have a strong cash balance that takes us into 2027, through our phase 3 program, and allows us also to invest actively in our commercialization initiatives. As Steve mentioned, we see a more aggressive timeline now for our commercialization, and we are making the appropriate investments that we have to ensure that we're ready to launch when the drug is approved. And if the drug is approved, but we expect it. As you see, we've got a catalyst-rich 2026 ahead of us. So exciting times for Compass Pathways, the leader in psychedelic drug development, in a very exciting new area of science. So with that, let me hand the call back to Aaron at Edison to open the discussion to questions. Thank you.
Excellent. Thank you both. It's really interesting to see the work done by Compass there. I wonder, I'm sure our audience would be keen to hear a little bit more from the both of you. Maybe you could elaborate on some of the regulatory considerations when looking at psilocybin-based approaches, and maybe what could be interesting is touching upon how those considerations differ from country to country.
Steve, you want to touch on that?
Yeah, happy to start there. Of course, in what we've presented so far, we've mostly focused on the U.S. market. But that said, from the inception of Compass Pathways and the design of our clinical development program, we've designed this to meet regulatory standards not only in the U.S. but in many other markets around the world. Many of our trial sites have been in the U.K. and the EU. We have taken early scientific advice in the EU. And we do intend to file there and in other markets as well. As far as regulatory considerations in the U.S., the FDA did put out draft guidance in June of 2023 to guide sponsors on the design of an acceptable development program for psychedelic treatments. If you look at the design of our phase 3 program, you will see that it overlays that guidance very nicely. And so we believe we do meet all of the standards that FDA has laid out. And probably one of the most important takeaways from their guidance is that they expect the same standards to apply for the investigation and development of psychedelic treatments as for any other treatment that's being developed within psychiatry or across medicine. So we really have taken pains to really design rigorous trials that will withstand scrutiny and adequately demonstrate the safety and efficacy profile of COMP360.
Thank you very much. We've had a question come in that sort of says that there's several companies developing psychedelic therapies for mental health conditions. It'd be great to hear what makes Compass distinctive from other approaches in development. And could you also maybe touch upon how the competitive landscape, how you see that evolving in the coming years?
Yeah, should I start
Sure,
there, Steve?
Steve, you want to take? Yeah.
Yeah. I mean, first, I think it's worth saying that it's almost silly to think about a competitive landscape per se, given the size of the unmet needs across a range of mental health conditions. Again, more than 3 million U.S. adults living with treatment-resistant depression, 13 million with PTSD, and there are comparable unmet needs across many other mental health conditions. There are many other companies that are developing psychedelic compounds of various types across a range of mental health conditions. There are fewer that are also developing treatments in TRD or in PTSD. But there are other programs in generalized anxiety disorder, major depressive disorder, etc. In terms of development progress, we are certainly well in the lead. We would expect, if approved, to be the first to come to market and by a fairly wide margin. There are a couple of other companies developing treatments specifically in TRD that are fairly far behind us. A couple of those are in 5-MeO-DMT. Ultimately, we think of this not as a winner-takes-all environment, given the size of the unmet need. This is a patient population that is going to need many new options, many new tools. There may be some differentiation ultimately in terms of the patient experience, the provider experience of delivering these treatments, potentially some of the economics associated with delivering these treatments. And it's partly to better understand all of that, as well as to ensure the rapid uptake into the healthcare system of our treatment if approved, that we've done so much pre-commercial work with our strategic collaborations, which represent a variety of sites of mental health care delivery, including interventional psychiatry clinics and hospital systems, integrated delivery networks, community behavioral health, and more, to really understand what are any potential barriers to the implementation of COMP360, if approved, to be delivered by healthcare providers. And we are very confident at this point that there is an existing infrastructure that is ready, willing, and excited to deliver COMP360 as soon as it's available.
I would just add to that, as Steve mentioned before, Spravato is really the only treatment that is utilized today for the most part in treatment-resistant depression. And as we look at the competitive landscape, we don't really look behind us, as Steve mentioned. We really look at, what treatments are being utilized today, and what are the shortcomings of those treatments. And what we see with Spravato is that it does lack a durability. And the data that we delivered in our phase 2 and our first of 2 phase 3s resulted in MADRS reductions, depression score reductions that are consistent with the depression scores that were seen in the Spravato application, the new drug application. So consistent. However, it took Spravato 8 treatment visits to have the equivalent reduction in depression score that only one administration of COMP360 provided. So we think there's quite a significant advantage for COMP360 over the only treatment that's really being used right now in the market.
Excellent. Steve, thank you very much for the insightful discussion. I think in the interest of time, we're going to have to move on to the final talk of the day, but thank you very much. Our final talk then is with Newron Pharmaceuticals, where we have CEO Stefan Weber. Stefan is the Managing Director and CEO of Newron. He's raised several $100 million in funds for Newron and his previous biotech companies, including another IPO. So Stefan's talk today will cover the current unmet needs in schizophrenia and how Newron is developing evenamide as a potential solution. So Stefan, the floor is yours.
Thank you, Aaron, and thanks for taking us. So Newron is 26 years of innovation in CNS diseases. And as you might expect, after 26 years, we do have a drug on the market already, which is an add-on treatment for Parkinson's disease. It's globally licensed. We have collected about 100 million revenues in royalties and milestones, and that is nicely growing at a double-digit percentage year by year, and will go on for a number of years. So it's a nice contribution to the operational cost. But first of all, it's a proof of the pudding that we know how to take drugs to the market, that the class of compounds, because the schizophrenia compound, which we will focus on today, comes from the same class of compounds. These compounds are extremely safe in man, and finally they work in the brain. So Newron is a small team, headquartered in Milan, Italy, with a subsidiary in Morristown, New Jersey. And we are listed on the Swiss Stock Exchange for 20 years, clearly with ambitions now, as we are in the pivotal stages of our schizophrenia drug, to have our shares listed as well on the NASDAQ. Up to the next slide, please. Thank you. So let's have a look at what the schizophrenia market looks like today and what is the real need that needs to be taken care of. In fact, schizophrenia is with mankind as long as mankind exists. We are looking at 1% of the population around the world, and that means we currently have about 25 million patients actively treated. This disease starts in males and women in their late teen and early 20 ages, and it is going to be a lifelong disease staying with them until they die. Schizophrenia patients will die about 10 years earlier than the average patient. The average person, and treatment-resistant schizophrenics will die about 20 years early. And that is after a lifelong career of schizophrenia. We know positive symptoms, which are those like having hallucinations, hearing voices, signs of grandiosity, walking in the streets. We have negative symptoms, people who are sitting in a room and looking at a wall for hours without emotions, and we have the cognitive decline. The cost of this disease to the society is way beyond the simple treatment with drugs. If we take the direct cost in the US only, that's $63 billion every year. Now, I would lie if I said, well, ours is going to be the first treatment. There's schizophrenia treatments on the market for more than 60 years. And unfortunately, I will say they all go back to an invention by chance when a doctor tried a dopaminergic mechanistic drug in a pre-surgery treatment of an agitated patient. And since then, with two exceptions, I would say clozapine and the late Karuna drug Cobenfy, all drugs in between have been more or less copies of that one invention by chance. And that means they all work by dopaminergic and serotonergic mechanism. And unfortunately, those drugs do some good to most patients initially, like 70% of patients who have their first episode will benefit from the current dopaminergic, serotonergic drugs. But as they go on, they will go step by step into that downward spiral for the rest of their life, which will make most of them, the vast majority, poor responders and treatment resistant. There's a large study, the so-called CATIE study, which says that after about 5 years into the disease, patients, and that is 72% of patients, will switch their medication every 18 months. And that is because either the drugs wear off on their effects or the side effects are simply unbearable. And we are talking about pound weight gain, sexual dysfunction, hormonal changes, and as we are playing around with dopamine, Parkinson-like symptoms. So our treatments today for schizophrenia are far, far, far from perfect. And on top, the efficacy is limited time-wise, and none of the drugs today covers negative symptoms and cognitive decline. So there is high need for something with a completely new mechanism to support what we have today in the armatorium of the doctors. And there's also a very high need for a first add-on treatment. The simple reason why we don't have add-on treatments is that if all drugs work by the same mechanism, add-on means increase of dose, and there won't be any additional benefit, but there would be additional side effects. And that is why FDA has never approved an add-on treatment. I spoke about two treatments being out of that box of all the same. One is clozapine, which is a magical drug for the about 1/3 of patients in which it works. The best effects ever, but this drug is potentially deadly because it tends to kill white blood cells, causing sepsis and killing the patient. That is why only 5% of the US patients who by official guidelines should take clozapine will take clozapine, and 95% of patients will be treated off-label. And in Europe, those numbers are 15% will take clozapine as per the guidance, and 85% will be treated off-label with whatever the doctor believes is a medication for them. We know for a number of years now that the answer to an additional or better treatment paradigm might be glutamate. Glutamate is one of the most prevalent neurotransmitters in the brain, but it's extremely tricky to work with because if you reduce or increase it indifferentiately, you have massive load of side effects. In fact, there's only one example of a glutamatergic drug that has been commercially successful, and that's J&J's esketamine, which is doing $1 billion in depression. Let's move on to the next slide. So the key question is, why do today's drugs only give limited time-wise and range-wise benefits, and why do we have that load of side effects, and why don't we get closer to a better treatment for our 1% of the population? And there was exciting research done by Pittsburgh University and disclosed peer-reviewed in August of this year. And what Pittsburgh University says is, look guys, for the last 60 years we might perhaps have missed the target because all today's drugs target the dopaminergic pathway. And that is about 4 levels below where we see the origin of schizophrenia. And Pittsburgh University, Dr. Grace has said, look, the way we see it and the work we have done shows us that schizophrenia originates at the hippocampus, which is a part of the brain where social interaction mostly is driven or started and ruled. And what he found is that if you see hyperexcitability in the neurons of the hippocampus, which trigger positive symptoms, and negative symptoms and cognitive decline, he says that a treatment that really takes care of the key issues of schizophrenia should target the hyperexcitability at the hippocampus. While today's drugs should not be expected to do anything good on negative symptoms or cognition, because they don't even get to the origin of the disease and they don't target it. Also by the fact that these drugs today are working 4 steps down in the pathway, they have widespread side effects which are already mentioned. Now let's talk a second about Cobenfy, because Cobenfy claims that it's a cholinergic drug, and I won't dispute it, and that it's a completely new mechanism of action. And that is why Bristol Myers, who acquired Cobenfy for $14 billion, as a revolution in schizophrenia, did a phase 3 study as an add-on treatment in schizophrenia. And the study results, unfortunately, were negative in July, which again confirms that if you take one of those drugs today, dopaminergic pathway, and you add it to another dopaminergic drug, again at the wrong target in the pathway, you won't get improved results. So the message is, let's find a new mechanism of action that takes care of the hyperexcitability at the hippocampus. And that takes us to our compound evenamide. Next slide, please. This one, yes, thank you. So what our drug does is very simple. It's a voltage-gated sodium channel blocker. I call it a magic switch. My CMO will say, what a bullshit. It's all science, but magic switch explains it nicely. This drug does only act if, when, and to the extent required. So it's a light switch, like I turn on if I need, I turn off if I don't need, and then there's the dimmer in between. How does this drug do this? This drug is only closing down nerve cells that fire excessively. Those nerve cells that are firing excessively can be silenced with a little drop of drug. While all the nerve cells that are firing normally, you need a bucket of drug to impress those nerve cells. And that is what you see in the middle of the chart. So excessively firing nerve cells are shut down, while normally firing nerve cells are kept active. And to the right, you see how that translates into chemistry in the brain. We are looking at the glutamate levels in the brain. And you see to the left of that right chart, you see all 3 curves being identical. That means that in a normally firing hippocampus, normally firing nerve cells, our drug will not have any impact on basal level of glutamate, and therefore we don't have any side effects. The magic switch. But if we trigger excessive release of glutamate, like in the black line, then we have an immediate and dose-dependent modulation of excess release of glutamate in the brain by our drug. When it comes down again to normal levels, again, we don't impact the basal level. So the message is, our drug does dose-dependently modulate excessive release of glutamate. And as you see in the next chart, it does so exactly at the spot where it is required, because as per Pittsburgh University, our drug normalizes neuronal activity at the hippocampus, it improves cognition, and it improves negative symptoms, all in the work done by Pittsburgh University. And there's one thing which will prove to be very exciting and important for the next slides, and that is that the Pittsburgh University looked at the time of the duration of the effect of our drug. And excitingly, even if our drug is degraded in the body after 20 minutes, no longer than 30 minutes, any presence in the body, there's also no metabolites, there's nothing active left. The effects of this drug goes on for hours. And that means the only explanation is we have neuroplasticity in the brain caused by our drug. So let's look at where we are with our drug. We do have the first ever 1-year study in treatment-resistant schizophrenia. That's 30 to 50% of the population. And we have been adding our drug to each and any antipsychotic of relevance in the world, excluding clozapine in that phase 2 study. What we saw, in line with what the prediction was by Pittsburgh University, we saw ever-increasing efficacy measured in the PANSS total mean change. We saw doubling and tripling responder rates on all the other endpoints. And what we saw after 1 year was that 50% of patients no longer qualified as treatment resistant. And on top, we saw something never reported before, that 25% of patients were in remission, so free from any relevant symptoms for a minimum of 6 months. So these results were kind of impressive. And the KOL said, well, if this can be confirmed in a double-blind, placebo-controlled international study, that would really be a potential change to the treatment paradigm in schizophrenia, because we would have an efficacious, safe add-on therapy in those patients who do not respond to any medication. Just to add one point, we didn't have a single relapse in 161 patients in that phase 2 study over 1 year. Okay, so a few months later, we had results from a double-blind placebo-controlled study, phase 3 study in 11 countries in patients who are poor responders to antipsychotic treatments. And this time, this phase 3 study included all antipsychotics of relevance, representing about 90% of the global market, including clozapine. And this was a 4-week study only in 291 patients. And what we saw was that we had highly statistically significant outcome on the PANSS total, the primary endpoint, a p-value of 0.006. We had highly statistically significant outcome on the secondary outcome measure, that was the CGI-S, Clinical Global Impression of Severity. We had highly significant outcome on all the other endpoints, responder rates, whatever you take. Excitingly, what we saw is that we had no relevant side effects. The most prevalent side effect was nasopharyngitis, a tripping nose. Compare that to 40% GI side effects in Cobenfy, and that load of side effects from the current antipsychotics. This study, most importantly, improved patients on each and any background medication. And for the first time, a company was able to show that its antipsychotic, when added to clozapine, could improve the clozapine patients. And olanzapine, one of the 2 best-selling drugs in the world, my chief medical claims that he has never seen any drug do better than olanzapine. We improved olanzapine by 6 points on the PANSS total scale compared to olanzapine alone. So impressive, highly statistically significant results from a phase 3 study as well. And as I mentioned, the safety profile was absolutely clean. Takes us to the next slide. We have a first-in-class glutamate modulator with phase 2 and phase 3 results already. We are addressing the large underserved TIS population and the poor responders, in total about 70% of the population. And this drug can be expanded into bipolar depression. And importantly, as today's antipsychotics should not be taken by elderly and demented patients, this is the one drug that can be added also in those patients. Now, we have started, and I should mention that we have gotten a nice validation of everything, the mechanism, the positioning, the sales potential, and we have also gotten a nice indication of the intrinsic value of this compound by a deal that we signed with the Eisai Group. We have heard the name before today. Eisai is a top 10 Japanese pharma company, has plenty of experience in CNS development. Lecanemab is their compound, and they also had another dementia drug before. So we signed a deal for 7% of the world market, Japan and other Asian territories, with Eisai. And what we got was up to 117 million euros, of which 50 million are already in our bank. That's important because that pays for our first pivotal study that I'm going to explain to you in a second. We got a validation of the mechanism, the positioning, and the intrinsic value of this transaction, if we discount all the future payments by risk and by interest, is more than 100 million euros as per Jefferies, who supported us in the deal. So if you want to do an intrinsic value calculation, end of December last year, 7% of the world market were worth more than 100 million euros. The money from that Eisai transaction has been used to kick off our pivotal program. The first pivotal study, Enigma TRS1, has been initiated. and is enrolling patients since August. Right now, we are enrolling patients on all 3 target continents. That's Asia, Europe, and America. That is going to be a 600-patient study in treatment-resistant schizophrenia. It will be a 1-year study. We will measure after 12 weeks if we can reject the null hypothesis, so if the drug works. Based on those 12 weeks readout, we will submit the NDA in Q1 2027. And given this is a high unmet medical need, we expect a 6-month review and the drug to be approved by end of 27. But we will not stop after 12 weeks. We will go on double-blind placebo-controlled until 26 weeks. We will measure the maintenance effect of that drug, which is important for EMA because they will waive the relapse prevention study if we are successful. And then for a lot of good reasons, pharmacoeconomic, we will go on for the full 52 weeks study. If this study confirms anything close to what we saw in the 1-year phase 2 study in treatment-resistant patients, this is going to be a landmark study for quite a while. But as approval of a new drug in treatment-resistant schizophrenia should be based on one sufficiently positive study. And the big word and the question mark is sufficiently. It's in the eye of the viewer, and we don't know what will be next October when we see the results from the 12-week. So the question with investors and the discussion with investors and pharma companies was, what if the reviewer doesn't think it's sufficiently positive, even if it's positive? So we said, look, if that drug is approved, it could be $1 billion a year. It should be, because all antipsychotics are. So we would lose $2 billion. What is the cost of a second study in comparison? And we did a 12-week study, and we did a 400-patient study, double-blind placebo-controlled global study. So we are going to start that second pivotal study in the very next few days with first-class academic US study centers. And you will see the press release sometime next few days, because we are still fighting with that first-class institution on the wording of the final agreement and the press release. That study will report results of 12 weeks in October next year as well. So, assuming that we have 2 pivotal studies reading out October 26, we have 2 shots at target. Each one of those 2 studies, if sufficiently positive, will do alone. But if both are just positive, that will do as well. Then we have a completely new mechanism of action, the first drug for treatment-resistant and poorly responding patients, and the first add-on therapy in schizophrenia. So we are very much looking forward to next year. And I think that is our deck. Over to Aaron.
Stefan, thank you very much. We're going to jump straight in with an audience question we've had pop up just here, but I think you've just covered it at the end there. The question was, when will the Enigma 2 study start? It sounds like in the coming weeks. Is that correct?
I hope it is more hours than days. Look, we are not working with any commercial centers in the US. We are working with first-class academic institutions, real high reputation institutions. The downside is they are not as sophisticated in negotiating agreements and getting them signed and reviewing press releases. And so I'm confident we get that done, but that is all that's missing. It is a question of very few days, I hope.
Sounds good. Thank you for the updates. So I suppose my question to you is, maybe you can help our audience understand how evenamide is differentiated versus Cobenfy. I mean, you touched upon it just there. I wonder if you could elaborate a little further.
Yeah, I mean, evenamide, if we look at the mechanism, it's the only drug that works by modulating glutamate. That's number one. So if we understand that dopamine doesn't cut the pie, and there's lots of evidence that first episode schizophrenic patients have an overshoot, an excess of glutamate in their brain. So that's a huge promise for a next generation. And remember, we've just submitted a new composition of matter patent, which would, if granted, give us until 2044 the substance patent. So that's enough time to find out if evenamide might also work in very early patients, first episode patients, and perhaps we could stop this cascade, this downward spiral at the very beginning. But that's hope and for the future. We know this is the only drug that works by glutamate. There's no competitor left nor right. There is no other company that has tried in the last years to go for treatment-resistant schizophrenia. The last company was Lundbeck, who failed about 6 years ago. Nobody has touched it. And there's no good reason for it, because all the current mechanisms do not help the patients. There is simply no dopaminergic imbalance in poor responders. There is no dopaminergic imbalance in treatment-resistant patients. What we have is an excess release of glutamate. So our drug has the right answer scientifically. Commercially, there is no competitor. We are the one add-on therapy. If we were a monotherapy, we would fight against all other drugs, all other 30 drugs, which mostly are out of patent. That explains why Big Pharma pays 14 or 15 billion for our peers, one product companies, because they are desperate to get new pipelines built together. So we do not compete with any monotherapy. We are the only drug that will have a label to be an add-on therapy to each and any other drug, whatever it is. And that was nicely shown in our phase 3. We work as an add-on to every drug on the market. It's convenient for the patient, the doctor, and for society. The high risk of relapses, hospitalization, and increased suicidality is gone because you stay on your medication, you add evenamide, you can stay at home. That is why in none of our studies we have to hospitalize the patients. We are doing all our studies without patients, which is also nice for us because the studies cost only 1/3 or 1/4 of the studies of our competitors who go for monotherapy. But the most exciting thing is this: if you want to approve a monotherapy, which comes with a dopaminergic mechanism, that's easy going. You take a patient with schizophrenia who is on a monotherapy dopaminergic drug, which works. You take off the drug, you give your drug, it works. Well, bingo, you got a drug approved and $1 billion sales. What we do is slightly more challenging. We keep the patient on the best medication the doctor can give him. Real life, as more than 70% of patients who are desperately looking for a new treatment paradigm. We keep them on their drug and we add our drug on top. They get the benefits and no incremental side effects. And then once approved, the doctor can reduce the background medication. Because we have a low dose, we have a high dose, he can start with low dose evenamide, go up to high dose, reduce background medication, get rid of all those terrible side effects. That is the promise of evenamide.
Fantastic, thank you, Stefan. We've had a couple more questions come in, which I think are somewhat linked. First part of this is, will the upcoming press release only talk about Enigma 2, or is there more to come? And I think that follows on nicely with the next question with, how will this be financed? Will there be another licensing deal announced?
Yes. So I mentioned that the money from Eisai is enough to pay for Enigma TS1, and that's funded to the readout in October next year. The study centers in the United States that we are starting is funded by that money as well. In order to expand the second pivotal study to its full size, we are currently talking about a fundraise of 25 million euros. And given that we have submitted all the applications for expanding the second study globally, that will take about 3 to 4 months to get approved. So we have about 3 to 4 months to get those 25 million euros. And there's 4 options to be absolutely straight of how to raise that money. The simplest one is to sell the outstanding Xadago Parkinson drug royalties. I mean, this is low-risk business. The royalties are safe. They are growing double-digit. There will be no huge risk discount on that. The second option is to sign additional license deals like China, India, LATAM, and ultimately perhaps even Europe. The one territory we will not sign up before we get the pivotal results is the US, because we have seen from Karuna and Intracellular what the value of the US rights with an innovative schizophrenia drug can be. The next option is that we sign some future revenues, that we sell some future revenues of evenamide. And the last option is we go for an equity raise. Now, equity raises, we have enough shares to do it, but equity raises in our space are still very special things. If you look at LB Pharma, which was an IPO with a schizophrenia drug, they sold themselves away in the IPO for like $15-20 million pre-money, phase 3 ready. I cannot do that deal, I won't do that deal. And if you look at Minerva, which has just raised 80 million euros or dollars, I don't recall, and up to $200 million, in that case, there were lots of warrants added to it. So if you want to have big money in our space these days, even if you are phase 3, it is still challenging and very expensive money. And I am not sure I want to let my shareholders pay that high price. There's other sources of funds, as I explained to you.
Thank you, Stefan. Maybe just one quick final question, if I may. The final slide you had there sort of talks about potential expansion into other neuropsychiatric and neurodegenerative conditions. Could you maybe just give us a little bit more on what that might look like?
Well, you need to know that any schizophrenia drug that's approved will also be working in bipolar, depression, and in our case, in dementia, which is not open for the other drugs. And that leads to the situation that the market leaders do about $1.5 billion in schizophrenia, but the total sales are about $6 billion. If you look at intracellular, the real excitement in that drug, after being launched as a schizophrenic drug, the real excitement came with the bipolar patient group, the patient population, because that's higher priced. And that is why intracellular in 6 years was able to do $680 million peak sales in the US. And that was Intracellular, a biotech company who launched the drug themselves. They didn't sell it, they didn't license it. So Intracellular for us is the blueprint of ultimate success. Because after 6 years and having done $680 million of sales, their market cap was $9 billion. 16 times sales. I mean, even in pharma, you pay 5 times to buy a product, but they were worth 16 times their sales. And then comes J&J and pays another 60% premium to buy them for $15 billion. Well, if I can do that for my shareholders, I'm sure they will all like me for some while, at least.
Fantastic. Stefan, thank you very much for your time today.
Thank you, Aaron.
Thank you. That brings the event to a close. So thank you very much to all of our presenters for the highly insightful talks. It's great to see this range of exciting science, promising clinical programs from companies at the forefront of developing next generation treatments for patients with CNS conditions underserved by current options. Thank you very much to our audience also for joining today and for your questions. I hope we've covered them all, but if we missed any, please do feel free to reach out via email. And if you haven't already, do take a look at our thematic report on the CNS Renaissance on the Edison website. A recording of this webinar will be made available in the coming days. With that, I'll close the event and wish everyone a good afternoon. Thank you.